All right, so now we're going to look a little more closely at the synaptic cleft and transmission across that. All right, so synaptic transmission. Remember, the synapse is the space between the axon terminal, or synaptic knob, some call it, of the presynaptic neuron and a postsynaptic neuron. All right, and so that's what we have here. This is your presynaptic neuron, and this is your postsynaptic neuron. Remember that potential will come in, or there'll be something that will trigger these vesicles here, these synaptic vesicles, to release their contents into the synapse via exocytosis. Okay, and so you can see here we've got a voltage gated calcium channel involved in there, and so there's some different mechanisms and some different contents in the synaptic vesicles, some different neurotransmitters that have different purposes, right? And so the effects of those, of course, are going to be different. And over here on the postsynaptic neuron, you can see we have some receptors that are going to be specific to these types of neurotransmitters that are going to be released. So some neurotransmitters, remember, can be excitatory and some can be inhibitory. So some can open ion channels for a response to turn something on, or they may close an ion channel in order to turn something off. Chemically gated ion channels respond to those neurotransmitters then and cause a synaptic potential, and then that is going to be converted to a voltage change, which can then cause an action potential. All right, so synaptic potentials, I've already said they can be excitatory or inhibitory. So synaptic potentials can cause the cell membrane to depolarize or to hyperpolarize. And the sum of these together can determine whether or not a threshold is met and that action potential is triggered. Summation occurs at the trigger zone, which is usually the axon hillock. So excitatory postsynaptic potentials are graded. They will depolarize the membrane of the postsynaptic neuron, and the action potential of that neuron will become more likely. So excitatory increases the chances of an action potential occurring. Inhibitory can also be graded, that means summed together. They can hyperpolarize the membrane of a postsynaptic neuron and make it less likely for an action potential to be fired. So these excitatory or inhibitory potentials can be summed together and added together and lead to a greater or lesser probability, and summation is going to occur before you get to that, or right at that trigger zone, because remember, we have to have one at the threshold amount. And so if one is more positive and one can make it more negative, it's kind of a net effect. If the net effect reaches that threshold potential, one will be triggered. So we have over 100 different neurotransmitters just residing in the brain. We have peptide neurotransmitters that are made in the rough ER of a neuron cell body and transported in those vesicles down the axon to the terminal, to the axon terminal. Other neurotransmitters are made in the cytoplasm of the nerve terminals and stored in vesicles there. These here on this page are pretty important. You need to know these. You may have heard of some of these before. Acetylcholine you've probably heard of before. Norepinephrine is the same thing as noradrenaline. So epinephrine and adrenaline, norepinephrine and noradrenaline. Dopamine is one of those feel-good chemicals. Serotonin, again, is one of those feel-good chemicals. Histamine is related to allergies and things. And there are some others. So read through this list and try to kind of be familiar with these and what they do. All right, so let's review these events leading to that neurotransmitter release. All right, so the action potential passes along an axon on down to the synaptic knob. the synaptic knob becomes more permeable to an ion like calcium ions, and they diffuse inside. In the presence of calcium ions, these synaptic vesicles fuse to the synaptic membrane. The vesicles here then fuse to that membrane and release their contents into the synaptic cleft via exocytosis, and then synaptic vesicles become part of the membrane, and the membrane is going to give the material for the endocytotic vesicles. Neuropeptides. Remember, a peptide is a protein. We have neurons in the brain or the spinal cord that make neuropeptides that can act as neurotransmitters. Other neuropeptides can act as a neuromodulator. And what does it mean to modulate something? Well, that means to help control it. So these can alter the response of a neuron to a particular neurotransmitter, or even block the release of a neurotransmitter. Some examples of these are enkephalins, beta endorphins, and what we call substance P. Enkephalins are present throughout the brain. They're also in the spinal cord, and these actually relieve pain, and they bind to the same receptors in the brain as morphine. These are opiate receptors. And so understanding this helps us to develop things like pain medications, understanding how these work. So beta endorphin is morphine-like. It is in the brain, in the cerebral spinal fluid, and it is also a pain reliever, but it's a little bit longer acting than the enkephalins. Substance P is 11 amino acids together and found throughout the body. It transmits pain impulses to the brain and the spinal cord. Enkephalins and endorphins, we believe, relieve pain by inhibiting that release of substance P. And so when we are able to mimic these when we're able to mimic these substances in inhibiting things, then that is how we're able to block the sense of pain and use things to stop pain. So the way the nervous system processes nerve impulses and acts on them reflects the way that neurons and axons are put together in the central nervous system. We have what we call neuronal pools, and these are interneurons in the central nervous system that are organized together in a little pool together, like when you pull your money together, okay? So they work together for a common function to receive input and generate output. So if you're pulling your money together to go and, say, buy a lottery ticket, okay, it's a common function. So these can be excitatory or inhibitory, all right? We have convergence, and that is when a single neuron in a pool can receive input from multiple neurons. So it's taking a bunch of messages in. If they come from different parts of the nervous system, they can converge or come together and allow the neuron to sum those impulses from different sources. And then we have divergence. The neuron is going to branch at several points and can signal multiple neurons. So it can tell multiple neurons. One can receive multiple and the other can tell multiple. So one neuron can stimulate two and two can stimulate several others, etc. Facilitation is just when some facilitate or help the action potential occur by releasing certain neurotransmitters near each other, which can increase or amplify that response. All right, so here's an example of convergence. You can see you have two neurons here that are sending a message to one. So there is one receiving. So it's receiving input from several neurons. The impulses represent information from different types of sensory receptors, and this lets us collect and process and respond to information. Makes it possible for a neuron to sum those together from a different source. Divergence is just the opposite, except in both cases we are making the most of what we have and working together here, okay? So here we've got one neuron sending an impulse to several neurons at the same time, which can increase an impulse. That can be from a single neuron in the central nervous system. That could be amplified to recruit several motor units needed in order for a muscle contraction occur. So it wants to recruit all of those motor units at one time. All right, so amphetamines, this is a good clinical application. Amphetamines are medications like Adderall. That's something that's used to treat ADHD. It's a stimulant. Amphetamines bind to a transporter protein and then they're taken into the neuron. Once inside, the amphetamine is going to disrupt storage of the neurotransmitters like dopamine or norepinephrine. These guys work to regulate mood and behavior. I remember norepinephrine is like noradrenaline. Adrenaline pumps you up, noradrenaline brings you back down. And what they do is inhibit a protein, and that protein is this one here called VMAT. VMAT's job is to transport the neurotransmitters into vesicles so that they can be released into the synaptic cleft. So its job is to release dopamine and norepinephrine. So what amphetamines do is after they alter that protein, they make that VMAT actually run in reverse, which increases the release of dopamine and norepinephrine into the synaptic cleft. So instead of taking it back up, it's loading and dumping tons of dopamine and norepinephrine, which is why people get that pleasure response, because you're overloading your system with lots of dopamine. This increases the effects on the central nervous system, which affects the areas in the midbrain that are related to that pleasure feeling. It's also tied to the delusions that they have and the addictive effect. Okay, norepinephrine has an effect on the fight or flight response. It can affect mood, emotions, movement, etc. These are also include some of the reward centers. That's a reward center is where your brain says, hey, I like this. This is a good thing. Let's do more of this. Okay. And so that reward center is triggered. And so that's what's partially responsible for that addictive nature of some drugs, and particularly amphetamines. So that was an example of something that was harmful in a drug that uses the body's physiology. This is an example of a medication we have that uses the body's physiology. We take advantage of understanding this and we know how to use it to treat it. And that is selective serotonin reuptake inhibitors. Serotonin is one of those feel good. Dopamine and serotonin both are involved with the pleasure response and help you to feel happy and content. And they just give you a good feeling in general. And so selective serotonin reuptake inhibitors, that's kind of a mouthful. So these medicines are used to treat depression, anxiety, those types of things. And what they do is they block the reuptake of neurotransmitters like serotonin from the synapse. So what happens is you have receptors over here, and these are released, okay, and then after amount of time they're taken back up, okay, just kind of like we were looking with the amphetamines and VMAT, okay, except this is a different it was dopamine, this is serotonin. And so what they do is they also, if you block the reuptake, if you block the reabsorption, I keep losing my pen, reabsorption, then you're going to have more of them here in the, they're going to increase here. I'm trying to make some neurotransmitters here. They're going to increase in concentration here, and so you're going to, in effect, to get more serotonin because it's going to have a higher chance of fitting into these little guys here that help take it back into the body, okay, the receptors. All right, so this is kind of showing you what I was just talking about. Serotonin is released from the vesicles into the synaptic cleft here. And then it's going to bind to these postsynaptic receptors and be absorbed and used. Okay, whatever's not used then is reabsorbed back up here. And so SSRIs, or selective serotonin reuptake inhibitors, block the reabsorption of them back there. So you have extra serotonin in the synapse, and so it's going to accumulate and increase the concentration that it binds into this, okay? And so it makes you feel better and helps increase your mood. A lot of times people will have a deficit in the amount of serotonin. But this is also part of the reason, too, why a lot of these medications you have to wean off, because because your body gets used to that and gets dependent on it. So it's very important to follow directions with these types of medications.